thank you very much, Don, and hello to everyone. So as Don mentioned, the talk today will be looking at evaluating monoclonal antibody PK properties in pregnant versus non-pregnant mice. So we have a very interesting collaboration between MorphoSys, a world-leading biotech company based close to Munich in Germany, and Will Research. And if I can move on to the next slide. So to begin with, I'll start with a few quick reminders, principally for my benefit rather than yours. And the first is to look at the basic IgG structure. So there are identical heavy chain light chain pairs with three important components. There are two antigen binding fragments that have unique antibody-antigen specific relationship. And then there is the constant region composed of sections of the heavy chain pair only that is involved in receptor binding. This is known as the Fc region, and it is part of the IgG molecule that is central to our discussions today. Next slide, please. And secondly, I would just like to mention passive immunity, which is the process whereby there is a transfer of immunoglobulin during pregnancy from the mother to the fetus. Indeed, at term, total IgG concentrations typically exceed maternal levels in humans. However, as Don has already mentioned, simple diffusion across the placenta is not possible due to the size of the IgG molecule with a mass of approximately 150 kilodaltons. Therefore, a receptor-mediated transport system is required, and it is the Fc region of the IgG molecule already mentioned that binds to the receptor. And the receptor responsible for this passive immunization is called the neonatal Fc receptor, or FcRn. It is worthy of note that other immunoglobulins such as IgA and IgM lack the Fc region and are therefore not transported across the placenta. Next slide, please. Obviously, the transport system is found at the interface between the mother and the fetus, that is, the placenta. A placenta can be considered as any apposition of embryonic to parental tissue for the purpose of physiological exchange. Small molecules can freely cross the placenta via passive diffusion, generally throughout gestation. Conversely, large molecule biotherapeutics generally do not cross the placenta because of their size. However, antibody-like biopharmaceuticals that contain an Fc region like in IgG are unique because they can be transported across the placenta via the FcRn receptor-mediated mechanism. Next slide, please. I'm not going to go into any detail here, but clearly there are different placental types across the mammalian species. The definitive type of placenta seen in rodents, rabbits and higher primates, including humans, is chorioallantoic. However, as you know, rodents and rabbits have two placenta, the definitive chorioallantoic placenta and an earlier inverted yolk sac placenta. In primates, antibodies have been demonstrated to cross the syncytial trophoblast cell layer of the chorioallantoic placenta. So the questions are, where and to what degree does the transfer of IgG and Fc containing biopharmaceuticals occur in the different species, and in this particular case in rodents? Next slide, please. Obviously these questions have already been addressed, and one of the most cited publications in the literature, including in regulatory guidelines, is by Pensuk and van der Laan from 2009, an interspecies comparison of placental transfer, new insights into developmental toxicity testing of monoclonal antibodies. It's perhaps difficult to see this graph, but it shows fetal maternal IgG ratio during pregnancy in humans and in several animal species, including the primate, rabbit, guinea pig and rat. In general, there is an increase in fetal IgG levels during the third trimester, at least reaching equivalence with maternal levels at birth. However, the flat green line in this bottom right-hand corner of the graph represents rodent IgG levels. The authors of the publication stated that neonatal rodents, so rats and mice, receive passive immunity predominantly postnatally via the gut FcRn receptors, so this is via the milk. And they go on to say that maternal-fetal IgG transferred prenatally in rodents is low. Next slide, please. And this is where our work with morphosis comes into the story. It begins with an initial unexpected finding in a PK study by the IV route in pregnant mice with a rat IgG2A antibody against a mouse-specific antigen. So the test item was a surrogate for a human IgG1 drug candidate. This graph shows the mean maternal plasma concentrations against the gestation sampling day. The blue line shows the exposure we were expecting to see based on previous in vivo studies in male and non-pregnant females with the same antibody. However, what we saw was an enhanced clearance rate from gestation day 9 until the end of pregnancy, as depicted by the red line. And this was the case for all animals with the exception of one female, which had a profile compatible with the blue line. And yes, you've guessed it, it turned out to be incidentally not pregnant. It was therefore no surprise that the first question from the study monitor was, had we dosed all of their animals correctly? Next slide, please. However, the initial finding was confirmed in an additional PK study using pregnant and non-pregnant mice. And other studies also demonstrated that the increased clearance was not a mouse strain related effect because we had the same finding in a second mouse strain. It was not an antigen mediated effect because the same finding was observed with a mouse antimurine antigen IgG2A antibody.